Alors bonjour tout le monde, bienvenue à cette conférence, ce séminaire du CEF. Mon nom est Jean-Pierre Tremblay, je suis directeur du Université Laval depuis septembre. Donc c'est le premier séminaire que j'ai l'occasion d'animer. C'est un grand plaisir parce qu'en plus c'est un séminaire de postdoc et vous savez que les séminaires de postdoc c'est les meilleurs. Toujours. Donc on a le plaisir d'accueillir aujourd'hui, pour me tromper de nom, Anne Holla. Qui vient de l'Université de Queensland, qui est présentement, actuellement, au doctorat ici, au département de chimie, à la faculté de sciences et génie. Elle travaille avec Florent Dominé, expert des relations avec la neige et le péridisol. Mais c'est pas ce dont elle va nous parler aujourd'hui, elle va nous parler de ses travaux de doctorat qu'elle a réalisés en Australie sur les mangroves et le blue carbon. On va parler un petit peu de blue carbon. Donc, je lui laisse la parole. Anne. J'ai oublié de dire, c'est vrai. Elle vient de Queensland, mais avant ça, c'est une citoyenne du monde. En fait, elle est allemande et elle a fait sa maîtrise à Lancaster Limited Time. Je cherche le nom. Grande-Bretagne. Royaume-Uni. Donc, c'est ça. Donc, pour s'en aller ensuite en Australie. C'est ce dont elle va nous parler aujourd'hui.
Okay. So, hello everybody. I'm going to talk today about the roots of blue carbon in mangrove forests. And this is the outline of my talk. So first, I'm going to start off with talking a little bit about blue carbon in general. Then I'll chat about blue carbon specifically in mangrove forests. Then after that, I'm going to talk about the mangrove forest, which is a very unique environment. And then finally, I'm going to finish off with the effect of environmental factors on mangrove productivity and carbon cycling. Now, blue carbon. Blue carbon is basically all the carbon that is associated with vegetated coastal ecosystems. And those ecosystems are tidal marshes, which you can see here, then mangrove ecosystems, which you can see on this picture, as well as seagrass meadows, which you can see here. Now, there have been many efforts to quantify the carbon that's stored in those ecosystems. And if you look into this column, you can see that the seagrass meadows really outperform mangroves as well as salt marshes in terms of carbon burial rates. However, these figures are largely due to, or this figure is largely due to the extent of seagrass meadows on a global scale. So if you take this into account, you will see that actually mangroves as well as salt marshes are much more efficient in sequestering carbon than seagrasses. Now, you are probably more familiar with carbon sequestration rates in terrestrial forests. So how does this compare? Well, this is a figure that shows, where is it again? Here. So you can see here how mangrove carbon sequestration compares to purely terrestrial forests, such as tropical forests, boreal forests, as well as temperate forests. So mangroves are really, really efficient in sequestering carbon. And I'm going to talk about the reasons for this in a bit. Now, blue carbon in mangrove forests. On this map, So on this map, you can see how it's distributed globally. And you can easily spot that there's a hotspot down here in Indonesia. This hotspot, in terms of total soil carbon stocks, is followed by Australia, Nigeria, Brazil, as well as Mexico. And those carbon stocks are largely due to the mangrove area in these countries. So you can see that, for example, Indonesia holds about 22% of the world's mangroves, and Brazil, Nigeria, and Australia hold about 6%. So it is really a matter of area in that case. However, in addition to this, I should probably mention it, that mangroves are mostly found, or they are only found in the tropics and in the subtropics, which you can see here on the distribution map. However, again, in terms of species richness, there is a hotspot in Indonesia. And so in addition to the hotspots in total carbon content, you'll find species richness as high as here. There are about 47 mangrove species globally. And a recent or fairly recent paper from Atwood et al. Tried to establish how important are species in these total carbon stocks. And they found that, for example, mixed forests are having significantly higher carbon stocks than monotypic forests, which is, well, I guess competition is somewhat beneficial. And then they looked into genus richness, which you can see here, and they found that if you have 5 genera, you will get the highest carbon stocks. And this is most probably likely due to, well, there are some genera are simply more efficient than others or more productive than others, which you can see here, but I will keep coming back to this graph as we go, yeah. Now, let's talk a little bit about mangroves. It is a very unique environment, and I chose this picture because here you can already see what's the main driver of the system. So the mangroves are located right between the land and the sea, and this means that they're regularly inundated by tides. And with the tidal inundation, there comes high soil salinity, as well as waterlogging or anoxic soils. Now, mangroves have a number of adaptations that allows them to cope with those conditions. For example, they are viviparous, they're salt tolerant, and some of those species have aerial roots. Vivipary. So vivipary is a condition that is found only in a few seed plants. It means that the seed develops in a seedling without dormancy, and all of this happens on the adult tree. Now you can see this is a little drawing, so you can see, well, this is the fruit and how the radicle extends. And then once it's ready, it will be chopped, and upon soil contact, you will have little roots developing as well as the shoot developing. Salt tolerance. Well, this is a vast topic in mangroves, and it's not exactly one of my core themes. However, there is no way of doing a talk about mangroves without mentioning salinity, because, as I said, it's a main driver in the system. Now, mangroves are obligate halophyte, which means if you have a glasshouse trial and you attempt to water them with fresh water, you will kill them. On the other hand, if you exceed tolerance limits, which are about 90 parts per thousand of sodium chloride, you will kill them too. But obviously, this number really depends on the species, so the tolerance limits vary. One way to deal with salinity is the maintenance of high intracellular water potential, as in most halophytes. And the other kind of fancy thing about mangroves is that they do seem to have, or they do seem to exclude salt at the roots. So the root system functions as an ultrafiltration system. We don't know much about this mechanism yet, but it seems, obviously, it's not 100% efficient in reducing salinity. So the salt that is that enters the plant, it may be excreted at the leaf level, which you can see here. So those are leaves of Avicennia marina, and the species, for example, has some glands where it releases the salt. And it seems that salt excretion is present in species that are less efficient in filtering out the salts at the root level, which is something you can see here. So here you can see that the concentration of the salt in the sap, and you can see that the higher that is, the more excretion occurs in the species. Now, the other species, the salt that enters the system, they may deal with it using or shedding leaves. Having said this, there's only one species that is deciduous that kind of chops all the leaves on a regular basis. The species is Xylocarpus mecongensis. The other species, well, they chop like a leaf or a couple of leaves at a time whenever, which you can see here. And obviously another mechanism is to increase the succulence, which is basically the water potential or the water content of the leaves, or to enhance growth, which feeds back into maintaining high water potential in the leaves. Now, okay, this was obviously just scratching the surface on salinity. It's a complex topic, as I said before, but we have to move on. Now let's talk about aerial roots. Well, aerial roots are only present in some species, and for this example, I've selected 2 different types of aerial roots. So you can see here, those are stilt roots. I chose them because they are associated with the most productive mangrove species, like Rhizophora and Laconcaria. And then also Avicennia marina, which has pneumatophores, those are the snorkel-like roots. And Avicennia I selected that one because it's one of the most common species all over the world. Now, those aerial roots, they are equipped with those white little openings, and these are called lentisols. You can see this here in pneumatophores, here, that's kind of a zoom in, and you can see them here. And they basically allow gaseous exchange with the atmosphere. And in addition to this, those aerial roots are often equipped with parenchyma, which is basically, the species then, or the mangroves then have aerial spaces or air spaces in the roots, and this allows the transport of oxygen as well as the storage of oxygen in the plant. Now, how do we do this? Okay. Now let's go back to our figure on total carbon stocks. Atwood and colleagues, they also, they found that carbon hotspots do not correspond to above count biomass production of those species that are present in those places. And this, if you think about the system, makes sense because with all the tidal action, the leaves that are being shed off, they may be exported out into the sea. So there is not much that can go into the soil. And those that may remain, there is a lot of consumption by crabs as well. And Karen McKee, she suggested, over 20 years ago, that it is actually that those carbon stocks are actually down to the root biomass. And again, this makes sense because you have a system where those roots, they provide direct organic matter inputs into the soil. And considering the condition, high salinity, very little oxygen, what goes in stays there. Decomposition is really inhibited. And this is how you end up, for example, in Belize, there are peats that have depths of about 10 meters. So there is a lot of accumulation happening. Now, but when I started my PhD, this figure came out afterwards, but what I started off thinking was, okay, so you have those different carbon stocks associated with different genera, and you can see that those root systems, so I tried to color this a little, you can see that those two species, they're associated with those kind of stilt root system. Then you've got Bruguera and Lumnicera, one of the Lumnicera species that are associated with knee roots. So those are poking out of the ground, they are little loops and they're easy to fall over actually. And then you got Avicennia and Sonaratia, they are associated with those snorkel-like pencil roots. You can see here the pneumatophores. And then on the other hand, you have this patch, or those species, or those genera, they are associated with buttress roots as well as including planket roots. And then you've got the weirdos down here, they don't have any aerial roots. Well, in fact, this one is a palm tree, but... So I was thinking, maybe the physiology above ground, there seems to be something going on, and maybe this is reflected, or there is a link between root physiology and carbon stocks, and that this extends below ground. Now, obviously, this is forming the genetic constraints, the frame. However, root systems are also highly plastic in response to environmental cues. Environmental cues such as soil resources, like water and nutrients, but also the density of a soil. And in mangroves, roots have been largely neglected. There hasn't been much work done on them. Neither has there been much work on the plasticity of those roots to any of those conditions. But what I found is this picture that kind of shows how stilt roots upon soil contact vary depending on substrate. So you can see here, this is one of the stilt roots that has grown into mud. You can see that this one is a stilt root that has grown into draining sand, and this is how it looks when it enters water solely. And this is obviously rather descriptive. I mean, it hasn't been the effects we can see here haven't been quantified. In addition to this, not in addition, but in the terrestrial literature, the effect of soil density has been much more studied, or there is exhaustive literature on it, in fact. And you can see, for example, that from this one study that an increase in soil bulk density from 1.0 to 1.8, what it does to roots of narrow-leaved ash. And, keep in mind the gradient in soil bulk density, because in mangroves, there's also a huge range of different soil bulk densities that occur there naturally. For example, soil book densities of about 0.12 gram per cubic centimeters have been reported in the US, whereas one of our field sites in Australia had a book density of about 1.37. And those high book densities are usually associated with, the mineral sandy soils, whereas the low book densities are associated with organic soils. And as I said before, there is this gradient in soil book densities that occur is slightly shifted. Again, this has to do with a high organic matter content in the mangrove soil, as well as the absence of agroforestry machinery that creates compaction of soils. Well, you don't get this in mangroves. Now, soil book density is actually quite important, and it's really complicated to study because of its interaction with all the other soil properties. So you can see here that I tried to summarize this quickly, and I'm just going to run through quickly. So book density has an effect on oxygen availability as well as water availability. There is an effect indirectly on temperature. There's also an effect of book density on temperature directly. All of this will feed into, microbial activity, and then there are flow-on effects on nutrients, and in the case of mangroves, even salinity. Now, all of those soil properties individually affect plant growth, but in terms of soil book density, what has been found is that above ground, you may get reductions in biomass, you may get reductions in the number of leaves, as well as in the height, whereas the volume of the stem tend to increase. In addition to this, if you look below ground, there are reductions in the length of roots commonly. There are increases in diameter. High density soils are often associated with surface rooting as well as lateral root proliferation. And there may be some effects on root anatomy, and this is something, what did I do? No. Okay, so this is something which you can see here. These are root histologies from narrow-leaved ash, and you can see that the xylem diameter is basically affected by soil book density. It's much larger in low book density conditions. And then there's also an effect on the CN ratio of the tissue. Okay, we did not know how the mangroves would respond, so I started off with a glasshouse trial, and variation in soil book density was achieved by changing the proportion of sand and perlite in the soil. So you can see up here that I studied 6 different soil book densities, and you can also see how the soil, or where is this one? No. Okay. So you can see how the soils differed, and I was mostly interested in the below ground traits. So I studied everything mineralizer would give me, as well as biomass. And in addition to this, I looked into those 3 above ground traits. Now, what I found was that, and I was working with the locals. So local to Brisbane is Avicennia marina, one species with pneumatophores. Then again, the Bruguera gymnorrhiza with the knee roots. Syrups australis with buttresses, as well as Rhizophora stylosa with stilt roots. And those are just 3 traits for the purpose of this demonstration. So you can see that those species responded very different. For example, this one, Rhizophora stylosa, it seemed to have a growth optima at a book density of about 0.4, as indicated by the peaks, which you can see here. And up here, you can see the root system. So this is a root system that had experienced growth conditions of about with a book density of about 0.4 gram per cubic centimeters, as opposed to 1.2 gram per cubic centimeter. So there was a huge difference. Now, Bruguera gymnorrhiza, Bruguera gymnorrhiza, this one responded somewhat similar, but not as pronounced. You can see that there's like a tiny peak at 0.6, another peak here at 0.4, and here again at 0.6. Avicennia marina, this one, there was a decrease here, and then bell-shaped responses in terms of the number of roots and root length. And Syrups australis, this one, flat lines, no response whatsoever. So the response really depended on species, and I've been studying 9 traits, 9 traits in total. So I was trying to find a way to summarize those growth responses. And I came across the plasticity index that had been developed by Valadares and colleagues. And it allowed me, well, I applied to the data and it allowed me to, to compare the overall plasticity. So what I found and what you can see here in this heat map is that Avicennia is really the most plastic species above ground, whereas Rhizophora is more plastic below ground. Now, having established that mangroves do respond to variation in soil bulk density, I wanted to do an experiment in the field under natural conditions that doesn't look into seedlings, but into mature trees. And that's actually quite difficult the way we decided then to manipulate soil bulk density, because I can't just use heavy agricultural machinery and drive through the forest. So what we did, we used those little root ingrowth bags, and I filled them with different soil bulk densities, the same bulk densities I studied before, so that I would be able to compare later on. And I studied, well, it's a bit messy. So there were two different study areas, the island here, North Strasbourg Island, and then the mainland. On each, there were 3 different sites. They're so close you can't see them here, but there were 3 here and 3 here. And this had to do with species distribution. So on this island, I basically studied Rhizophora stylosa and Avicennia marina. And here I studied Cerops australis as well as Avicennia marina. There were 5 replicate trees per species, per study site, and each of them was exposed to those 6 different ingrowth bags which I placed around the trunk of the tree. And this is what I found. So it seemed that increases in soil bulk density resulted in longer roots in all of those species, which was rather surprising. So I started looking into the causes for this, and I realized that, okay, we use those mesh bags, which kind of defines the size of the roots we were studying. So we were actually looking into fine roots. And fine roots, their function is to acquire resources such as water and nutrients. And obviously, those resources being limited in high-density soil, you would expect an increase. And then I started going through the literature again, obviously, and I found that rather recently there is accumulating evidence that people have observed this kind of pattern in, different species, terrestrial forests as well. So it really seems to be dependent, or the response really seems to be dependent on root type and function. On root type and function. And then the other thing I was thinking was that because, let's just go back to this map quickly. Now, this island happens to be the 2nd largest sand island in the world. And the first one, I don't know who's familiar with Australia, the largest island is Fraser Island. And this obviously, creates differences in background conditions. So this being sandy soil, high bulk densities at the baseline, whereas on the mainland, it's much more organic. So there are lower soil bulk densities. So what I was wondering, what happens if a root grows from obviously high bulk density into a root bag of 0.6, there would be a reduction, some sort of relief. Whereas if you turn this around and then you say like, okay, it grows from a 0.4, from a very low density soil, organic rich substrate into bulk density, the same soil bulk density, that would be an increase. So how does this play out? So I created this index that's basically just the difference in soil bulk densities they encountered. And it's actually a quite nice method to study foraging in general, like for in the field, under field conditions, because you can do split pot experiments and stuff, but this is something you could do in the field. And this is what I found. So you can see that the more of a density the roots had to overcome, or the more of an increase in bulk density they had to overcome, this is, here is where they didn't experience any. This would basically lengthen the roots. So that was quite exciting. Now, having realized that obviously function is a thing, root function, I then studied the stilt roots. So I constructed like many of those little containers here, and I replaced the soil underneath those stilt roots that were just about to touch or to penetrate the soil. And so you can see here how one of our experimental trees, I started off with 10 experimental trees, but I lost some in the process. And this is how the containers looked up on installment. And this is basically what I found. So you can see that after where is it again here? So after 5 months, I got, in very loose soil, I got very long roots, and in dense soils, I got very short but thick roots. And those effects were significant, which you can see here. But then I suppose in terms of blue carbon, it's really much more important what's, you know, the biomass is really the thing we are after. And we found that high bulk density soils increase the organic matter inputs. And then I was wondering, so what are the effects on, you know, on the anatomy as well, and then ultimately on chemistry, because all of this will influence the decomposability of the root material. And I prepared some histological slices, and you can see here that parenchyma tissue, for example, parenchyma tissue decreased, or the air spaces decreased with increasing soil bulk density, which could possibly explain the increase in the biomass. And we found something quite fancy when looking at those microscope slices, namely that high bulk density roots, they seem to have those bubbly features. And it took me some time to figure out what is this, or what this is, because there wasn't much in the literature about it, except for one paper where Tom Linson found that, or states that those are, or he called them fibrous strengths, mostly because they are consisting of lignified tissue, like the outside. We don't know much about their function. I suppose it could be to facilitate solid transport within the root, and maybe the lignified tissue to enforce and strengthen the root, but we don't know. There remains some research to be done. But yeah, it was quite exciting, and again, it got us thinking, so if it changes the tissue or the quality of the tissue, what does this do to blue carbon? How does it affect decomposition rates? Because of for my glasshouse experiment, I had heaps of root material left, and I put the root material into little litter bags and buried them in the soil at one of our field sites where it was incubated for about 3 months. And this is what I found. So soil bulk density increases in soil bulk density actually reduce decomposition rates, which was quite neat. And you can also see there is a difference in species. So the effect of soil bulk density is not as pronounced in rhizophora, which is this gray line, the one with the stilt roots. The decomposition rates in general, which you can see, for example, here, this is Avicennia, and this is syrups. And down here in this, let's say, second patch, you can see Rhizophora and Bruguera. They do align very well with those carbon stocks that had been estimated. So you can see that syrups and Avicennia are somewhat down here, and then Bruguera and Rhizophora are somewhat up here. And those decomposition rates, I'm going to skip that one. So those decomposition rates really align with lignin content. It's nothing new, but again, in the mangroves, not much has been done on roots, so it was kind of new again. And so you can see that Avicennia, so Avicennia had much lower percentage lignin content and higher cellulose content than the other two species. Now, and I think that's pretty much it. Let's just sum up. So soil bulk density influences mangrove root development. There are flow-on effects on decomposition rates. Species differ in a response to bulk density, as well as having different decomposition rates. And ultimately, it seems that high soil bulk density enhances carbon sequestration in mangrove forests. All right, thank you very much. Are there any questions?
Pour les questions, je vais vous demander d'une personne de vous apporter le micro, puisqu'on est enregistré. Et si jamais vous préférez poser votre question en français, je peux la traduire en broken English, pas de problème.
Thanks, Anna. That was really interesting results, really nice presentation. Do we know what are the factors that are influencing whether the soil bulk density in different mangrove soils, whether is it the sediments coming from inland, or is it just the underlying geology, and are most mangrove soils, like, is there a majority that are more organic compared to mineral, etc.?
We started thinking about this. We didn't have the time to look into it. It seems that obviously geology and deposition, sediment deposition, is a huge issue. Then close to river mounts, you get more organic material. Then it's currents as well. But it also seems that over time, the mangroves will also influence what's in there, and there is a change. So you would expect more organic matter to accumulate, especially if high wood density soils are more effective in sequestering carbon. So you would, well, but we haven't looked into this, like we didn't do any studies on that yet.
So there's a fact of how long the formation of mangrove has been there on...
I guess,
yeah,
I suppose. Yeah.
Nice.
Okay.
Hi, thanks for an interesting talk. I'm curious if there's a way to separate out whether the influence of the bulk density on the root development is biological versus physical. Because I would assume, resource availability and all those things that you highlighted would affect how root respond. But the fact that the bulk densities are different, the physical aspects, I wonder if that constrains the root development in some ways as well.
I didn't, so can you, what's the question?
I'm just wondering if you can tease apart the physical aspect of
the mechanical, like the
mechanical
effect versus
versus
all the, well, you could probably do some models, but I didn't have the data for it. I mean, in the glasshouse trial initially, what we did was I did monitor water content, for example, but what I found was that the water potential in the stem, above ground, in the shot, it was not affected. But then again, you have hormonal action kicking in that could just adjust it. So it's really difficult to tear those factors apart, even if you monitor all those things. So I did not succeed, but then again, it wasn't really the main focus of the topic. Or of the project, not the topic.
I have a question, a curiosity question. I know it's not one of your data, but maybe you know where it comes from. Well, you showed this graph at the really beginning on the richness, and the carbon stuff, sequestration.
Yeah, I know which.
Yeah,
the one.
And you have this. No, if you go back a bit or forward, I mean. Yeah.
That one.
So you have the genus richness, and you have this as the kind of results we all want to have. With
I wasn't involved in that study, just saying.
So do you have any idea why you have such a high carbon stuff when you reach 5?
I guess it has to do, and this is something you can see here. So, for example, in the Americas over here, where is it? Here. Yeah. No, but in the Americas, you only have 4 species or 5 species, all of them belonging to different genders. And yet, there are some of them, they have, for example, this Laguncaria, which has the highest soil, and I think it's about the species you get at the same time, and then, yeah, I think it's about the balance somewhat, but, yeah, okay.
Yes.
Yeah.
Hi, thanks so much. It's just a continuation of the question. And are those the 5 species or the 4 or 5 species in the Americas? Do they have different rooting patterns?
No, this is for example, let me find you this one slide. There are different species or even different genres, but for example, this one here, the one you can find in the Americas with the stilt roots, with those prop roots, is exactly like, it looks like Rhizophora, which won't, well, it's not called like this here, but it's basically, well, from my point of view, it's the same thing, but it's probably not. So someone who is into taxonomy would probably freak out now, but, so it's the same kind of above ground roots. It's just different biogeography and different species. But same high productivity, really.
Okay, and so the 5 different ones, do they all tend to have the prop roots?
No. So in the Americas, they have, for example, so in the Americas, they have this lacuncularia. They do have Avicennia, like the white mangrove, which is, where is it? Here. So they have Avicennia, and I think they have some sort of Lumnicera, but I've never really been in an American mangrove forest, so it's, there are similar species, or there, how do you call this? There is a similar set of species that they look very similar in Southeast Asia. It's just they don't belong to the same evolutionary process. Okay. This is, okay.
I have a question about the growth rates of different species in relation to the carbon stocks, or...
I can try. I don't know if I have the data on top of my hand, but if not, I could also send you an email afterwards if you would like that.
Because are the more productive species actually not producing as many roots, or are the more conservative species producing more roots and therefore more carbon stocked in the soil?
Well, where is this one slide? Here. No, it's not. It's not this one. I think it's, yeah, it was that one actually. So in terms of, so for example, you can see here that, like what I found was that rhizophora, that's the same that with the stilt roots again, like my data really matches that one in terms of, I got a lot of root biomass production in that species compared to all the other species. And the slow-growing species syrups is a very slow-growing species, and I think this one is not present in the Americas. But this species and the syrup response could also be because it simply didn't have the time to respond. But then again, I didn't include the root diameter data. It did respond in terms of root diameter. So yes and no, it could be, well, we don't know yet. Oh, this is okay.
Épuisez nos questions. So thanks a lot, Anne, for this very, very interesting talk. I was right at the beginning. Merci à tout le monde. Puis on vous invite à la prochaine édition qui est quand? C'est le premier séminaire, on sait pas? Après Noël peut-être? Janvier. Merci.